Here. What I'm going to do today is talk about the journal's perspective on how we review randomized clinical trial manuscripts, because my hope is that by sort of seeing it from the journal editorial review point of view, it'll do two things. One is it'll help you when you write a manuscript, and this applies, frankly, whether it's an observational study, a prospective study, an interventional prospective study. Applies really to all of those types. It'll help you prepare a manuscript in a way that and submit a set of documents that increases your chance that your submission will be fairly reviewed and positively received. The other thing that this might help you do is actually be a better peer reviewer. Look for things that journals care about so you can directly address them in your reviews, which helps with the validity of peer review and helps us publish good science and have a lower probability of publishing science that might lead to incorrect conclusions. These are JAMA branded slides. This is an adaptation of a talk that I give in my role as the senior statistical editor at JAMA. Megan, they're fine to post. Ignore the little copyright thing. It's just in the template. All right. So my disclosures, my academic affiliation is at UCLA. I work with the Research Institute there, and I am an employee of Berry Consultants. I always said that I was a special government employee for FDA. Who knows if that's still true? Um, and I am the senior statistical editor at JAMA, and we'll talk about that role. So this is the perspective of a high volume general medical journal. So JAMA is a part of a network of journals, just like Lancet and some others have networks of journals. It's published weekly, which means there's a lot of turnover and rapid review of things. And just to give you an idea of the sort of bottom line characteristics of the peer review process, there's 10,000 approximately submissions of something. That includes letters to the editors, editorials, all sorts of types. And of those, the overall acceptance rate's 12%. But a lot of those 10,000 are things that are invited, like an editorial to go along with a manuscript. So of the 5,500 submissions of research manuscripts, so these are generally unsolicited primary research manuscripts, the acceptance rate's about 5%. So in order to give good feedback and revise well the 5% that we ultimately publish, the peer review process is aimed at both making rapid decisions if the submission's not going to be published, and then carefully reviewing and revising and improving the submissions that have a meaningful chance. About half of what JAMA publishes are clinical trials. So I want at the very beginning to talk about two different areas of peer review. So the purpose of peer review is to figure out whether the manuscript should be published in the journal. It is not to improve your science. It's not to educate the author. It's simply to make a decision and then ultimately to help improve the quality of what is ultimately published. So it's based on the importance or impact of the study and its results, the scientific quality or rigor, and our attempt to not publish stuff that's not reproducible or invalid. And the effectiveness of the presentation of the material, the clarity, the balance, the transparency, and reporting. So meeting our standards for complete reporting, the good, the bad, and the ugly of what's happened with a study. And every study has all of those components. As a general rule, many journals divide their reviewers into content and methodology groups. The content reviewers are the ones who should be expert or near expert in the area of investigation. They, in a sense, are the peers in peer review. They assess the importance, the gap in the knowledge that's being addressed by the study, what the impact of the results, the likelihood it's actually going to change practice, um, and that there is a balanced and disciplined interpretation of results. Um, I'm actually not going to focus on this because this is an area of peer review you're probably most familiar with because you are all accomplished in your fields and you probably do content reviews, or if you shouldn't, we should figure out how to get you involved in this. I do want to point out the phrase likelihood of changing practice. At the high impact journals, that's a huge issue. So if you think your results should change practice, you should explicitly state that. The other piece of the content review before I move on to the methodologic review is this idea of a balanced and disciplined interpretation of results. And again, what I'm doing here is I'm trying to flip your usual perspective from that of an author to a journal editor position. So if we review a manuscript and the the data are potentially important, but the presentation of the results are very unbalanced. So an example might be a study in which the primary endpoint did not meet the criteria for being positive, but the authors write the entire manuscript based on a signal in a secondary or tertiary endpoint and talk about how wonderful it is that their treatment improves something. We have to then make the decision whether the data are so important that it's worth wrangling and cajoling the authors to reframe their entire report as being a negative study with an interesting tertiary finding. And if you write your initial uh, manuscript in a very balanced and transparent way, then we're much more likely to think that working with you will be a good experience and worth our time. And if you're clearly a zealot for an interpretation that's not consistent with the pre-specified design, we may decide that we're going to let another journal have the privilege of working with you. Um, so I want to tell you that really does make a difference. The methodologic review is focused on the methodology, the trial design, the integrity and fidelity of the actual conduct of the trial to the original design, data quality, were there issues with loss to follow-up, other sources of post-randomization exclusions that might lead to bias, um, and whether the analysis and statistical interpretation is loyal to a well-described pre-specified analysis plan. Now, if you don't have a statistical analysis plan that's clearly set out, we don't have a standard against which to judge the fidelity of your analysis. That's a bad thing because then we worry about sources of bias that I'm about to go through in some detail. So I'm going to focus on the methodologic review because that's both my area of responsibility and it's also something that I think is less transparent to most authors. So journals really care about complete, rigorous, balanced, and transparent reporting. And we think that's key to supporting evidence-based medicine and overall improving care. And the appropriateness and integrity of the statistical design is something that we focus on a lot, and all high-impact journals do. But there are things you can do from the very beginning of your study, through its conduct, through writing it up, through submitting it to the journal, and even in revision, that can increase or decrease your chance of successfully getting published in your journal of choice. And so we want to talk about those things. So as I mentioned, journals often have a separate statistical review process that's separate from content review that's focused on those things. So it turns out that many years ago, a very well-known research methodologist by the name of Steve Goodman, who's now at Stanford, did a survey. He's a lead statistician at Annals of Internal Medicine. And he was interested in whether or not journals actually did statistical review. And what he found is it was very inconsistent. His original work was then repeated in this work approximately 20 years later. And what they found is that when they surveyed, first of all, most editors didn't return their survey, so this is not rigorous work. but that about a third never used statistical reviewers. So they just assumed that their I'm sorry, that their content reviewers knew enough about research design and practice in their area to uh, identify issues with study design and conduct. About a third used it for some articles, and maybe less than a third used it for all articles. I will tell you that at the high impact journals at this point, if they're seriously considering your article, they're going to have a statistician look at it through some sort of mechanism, or you need to assume that they will. But it's interesting that it's still pretty inconsistent. So what is our review process? So you submit a manuscript, excuse me, to the journal, and all journals have some sort of what I'll call an initial triage process. At JAMA, it's reviewed by, I think it's now currently one of three senior or associate editors. These are people who take a quick look at it and generally assess whether it has a meaningful chance of being published if the peer reviews were positive. So there's two areas here. One is fit for the journal, and the other is overall quality. So fit for the journal is a huge issue. So for most things that you will have to submit in your career, there are journals that publish stuff like it, in which the readers are interested in the kind of work and results and populations that you're studying. And there are journals that just would never, that's just not what they do, regardless of the quality. And so you want to make sure you submit to a journal that publishes stuff like what you publish, or if it's something that's sort of so out of the box you're not sure, then there is a lot of value in sending an inquiry to the editor and asking if it's something that they would consider. Things that are a poor fit. So for example, JAMA doesn't publish case reports. JAMA doesn't publish very early phase investigations because we're primarily a journal read by clinicians. So things like that get submitted are just returned without review. And that's usually done within a couple of days to allow the authors to immediately resubmit to another journal and not slow them down by bogging it through some longer review process. Those that are not returned without review, which is the majority of manuscripts, and for some categories, the vast majority, are then sent simultaneously for content review and for statistical review. When those reviews come back, the handling editor, who's someone with domain expertise in the field, who's shepherding the manuscript through, um, makes a decision about whether it has a meaningful chance for being published. And they're not making a decision to, for example, request a revision. They're making a decision about whether the reviews are not sufficiently supportive or the overall picture is not sufficiently supportive to consider publication. And then they may just reject after review, provide those reviews to you. At both JAMA, and I know this to be true at New England Journal, but I assume it's true at many other journals, revisions are not requested without discussion in a larger group. Sometimes it's a few people, but for many of the research manuscripts, it comes to something called manuscript meeting. So twice a week, we have a 2-hour meeting where the manuscripts that have been reviewed and fared sufficiently well in peer review are discussed by a panel of usually between 15 and 20 associate editors. So this allows very wide-ranging input into the decision to request a revision versus reject versus refer to another manuscript within the JAMA family. And this is sort of where the larger medical perspective of, is this something that would be interesting to clinicians in general? Do we really think it will change practice? And often people outside of the primary area of expertise identify questions that are sort of obvious, but people were too close to it. And so there's a lot of good input in this. Based on the discussion at manuscript meeting, or for some manuscripts, discussion by a smaller group of senior editors, a decision is made to either request a revision or to reject after review. The vast majority of manuscripts for which a revision is requested, unless the authors are extremely uncooperative, will be accepted and published. So the primary sort of decision point is the request for a revision. When the revision comes, and I should say that the information sent to you when the revision is requested includes input from everybody who's involved in manuscript meeting, meaning if people made points there, they get captured and incorporated. So you're sort of getting the wisdom of the group, if you will. When the revision is submitted, the handling editor reviews the revision and basically judges to what extent the authors were able to respond to the questions, critiques, or requests for changes. And if it looks good, they may simply make an editorial decision to accept the manuscript at that point. For reasons of efficiency, we try to make decisions at that point and not send out manuscripts for re-review. I will just say I have a statistical manuscript for a completely different journal, a methodologic journal, that's now sending it back to me the third time as a reviewer for me to review, who just sends everything back to their reviewers. At JAMA, we try to avoid doing that. But if there's a question that the associate editor can't resolve, and that includes resolving with internal communications within the editorial staff or our statistical core, then they may send it out for secondary content or statistical review and make a decision. But usually what happens is the associate editor looks at it and a decision is made. Rarely will bring it back to manuscript meeting if there was something very contentious, or perhaps the authors in manuscript meeting, we thought the authors were going to be able to address something one way, but then when they respond, we find out we completely misunderstood the issue. They didn't have access to data that we thought they did, or vice versa. Okay. Sometimes we ask for further revisions. We do try to minimize that, although often there's sort of wording changes we want to make sure that the authors agree are correct. Okay. So this kind of review process is supported by a lot of people. So we have 5 statistical editors that have different areas of expertise. My area of focus is on clinical trials, but for example, Issa Dehabre at Harvard is an expert in causal inference from observational data, and the others have additional areas of different expertise. And then we have statistical reviewers that are even listed on the masthead of the journal that we use for the vast majority of our statistical reviewing during the first review. Again, they have different areas of expertise. And Valerie, I think, will be at our meeting, our face-to-face meeting. So what things get people into trouble? So these are the kinds of things that result in less enthusiasm for a submitted randomized clinical trial manuscript that are potentially avoidable if you can consider them throughout the process of design, conduct, and analysis. The first is the degree of pre-specification and the consistency of trial documents. I'm going to go into this in detail, but you want your grant, your design manuscript, if there is one, your protocol, your statistical analysis plan, and then what you actually report in your manuscript to all agree on what was the endpoint, how was it collected, how was it analyzed, how did you handle missing data, those sorts of things. You want the trial conduct, to the extent possible, and we all understand this is never perfect, to be adherent to the planned study design. And, areas of non-adherence are non-adherence to the assigned treatment, so so-called crossovers, dropouts, missing data, basically anything in which the behavior of the patients or the data collection, or I should say the trial staff as well, deviate from what you intended. This can be difficult to judge if there's not enough detail in the protocol. This is why it's important that you write a detailed protocol that would allow someone else to run your trial, meaning it has that kind of detail. It's also why you submit your protocol with your manuscript so that we can actually compare the protocol, what the protocol said you were going to do against what actually happened as reported in the manuscript. In data analysis, it's very similar. It's adherence to an adequately pre-specified analysis plan. This almost always requires a separate statistical analysis plan separate from the protocol. You just don't put enough details in a protocol. The statistical analysis plan that's in an NIH grant is, at the risk of overstepping, probably never adequate. I'm not sure I've ever seen a grant that told us enough about the analysis so we actually could judge whether the correct analysis had been followed. There are almost always situations in which one wants to do, or there are good reasons to do, unplanned post hoc analyses. That's okay as long as it is crystal clear which were the pre-specified analyses, which were the post hoc analyses, and that the relative emphasis or ordering of the two is kept consistent, meaning pre-specified first, post hoc last. So this relates to the presentation of the methods and results. You want the ordering of the methods. Our primary endpoint is this, our secondary endpoint is this, our exploratory endpoints are these. You want that ordering to be consistent in all your documents, your SAP, your protocol, your manuscript. You want to avoid any cherry picking where you sort of pick something that was really interesting and say, hey, look at this over the stuff that was uninteresting, unless that was the thing that was first listed. And then lastly, selected mention of non-significant differences. So it's very common with junior authors that the differences that were not significant that they didn't expect or didn't think were interesting, they just say these were non-significant, there were no significant differences in baseline characteristics at randomization. And then there's something that's equally non-significant that trends in the direction they like, and they say there was an observed non-significant trend of, X over Y. Okay? You can't be selective like that. You have to have the same threshold for reporting a difference, regardless of whether you like the difference or not. Okay. So I want to spend some time on trying to give you a graphical feeling for why it is we care so much about pre-specification and preserving the originally planned structure of the analysis. So the reason is that we're trying to avoid bias because the multiple ways that bias can creep into study conduct and reporting, all of which tend to be biased, not always, but tend to be biases in the direction of a positive treatment effect, are one of the root causes of the so-called reproducibility crisis. This idea that a lot of science that's published, if you just tried to do it again, you might get a different answer. Okay? So one of these things is bias from design decisions that are informed by knowledge of unblinded data. So if you, for example, were allowed to pick your endpoint after looking halfway through the trial at the differences seen in multiple different endpoints, you're going to cherry pick one that might be the most informative, or just might be the one that just happened to show more difference on that day by chance. And since those data would be included in the final analysis of the study, that would positively bias the result of your trial. So this is why DSMBs, who by necessity should see unblinded data with explicit labeling of treatment groups, never blind your DSMB, are particularly how should I say, they should be completely firewalled from making recommendations to changes in design unless it is to stop the trial for efficacy or safety according to pre-specified rules or to protect the safety of the research participants. But another source of bias is something that we call multiplicities. Multiplicities simply means you're testing multiple things. You're asking multiple questions. And the multiplicities we worry about are not the ones that are laid out in the statistical analysis plan. So recently, some of us were discussing a manuscript, excuse me, a publication. And in the publication, they had 3 different ways of looking at similar endpoints, and they had a particular procedure for controlling the overall risk of a type 1 error or false positive conclusion by using increasingly stringent criteria for statistical significance because of the multiple endpoints. It wasn't Bonferroni, but it was Bonferroni-like. That kind of multiplicity is fine because it's transparent and there is a plan for addressing the risk of a false positive result that can be then reviewed, critiqued, questioned, and judged by both the statistical reviewers and readers ultimately. What worries us are hidden multiplicities. So picture, for example, a trial in which the primary analysis, when it's run, misses significance. So analysis 1, which was the pre-specified analysis, gives you a result that's between null and significant. But it turns out that if you look at a different endpoint or you adjust for different variables or exclude the people who are enrolled on Thursday, you can actually get a significant result. Think of it as, you can think of it as trial by error, or just you look at analysis 1 and you think, I should have adjusted for this covariate or something like that. And it may turn out there's even a third way to analyze the data, where once you have the data in hand, you can find it and get a really impressively statistically significant result. The thing that worries us is that analysis 3 may be the only analysis that we as the journal reviewers see, right? We don't know what you do in the secrecy of your office. So we have to depend on documents to assure us that there are not analyses 1, 2, and 3, but the analysis that we see was the single pre-specified one that was defined without knowledge of the particular data that arose within your study. So another way that this happens that's less egregious than just hiding your first 2 analyses would be one in which you have multiple endpoints. So picture a study in which you have a primary endpoint, which is not significant. You may notice I put the dots in exactly the same place. You have a first secondary endpoint that is significant and a second secondary endpoint that's really significant. Sometimes what happens is when you read the text in the manuscript, it just sounds like that last endpoint was always the most important secondary. Notice how I've switched the ordering of the secondary endpoints, okay? And it's as if that was really what motivated the study. Yeah, we did look at mortality, but what we really cared about was the pain scale, okay? So this sort of selective reporting or selective emphasis, even if the report includes all the endpoints, gives a misleading interpretation of the overall result of the trial. So we worry about that. So how do we judge whether or not these things have happened? Because this is part of a journal's responsibility to try to minimize how much of this gets into the medical literature. And I will tell you that historically, there is a lot of stuff published in the medical literature where what you're seeing in the article is in fact only one of multiple things the investigators tried, and you have no idea what the other ones are. Okay. So what we do is we look at the consistency of the trial documents. So you send us a manuscript, and the manuscript has these rough areas: how you define the patient population, how you randomized, treated, what the endpoints were with a specific ordering of the endpoints. how you were going to analyze them, and if there was an interim analysis plan, say for futility or early stops for efficacy, what those analyses plans were. If the only thing we were able to look at was the manuscript, we would just have to trust you, the author, that this was in fact the pre-specified plan. But in fact, there are usually, and as a rule, multiple other documents that are time-stamped before the manuscript was prepared or the data were available that give us an idea about what the evolution of thinking was over time. So the study protocol, which should have a version that is locked before first patient in, and then if you revise it, you need to keep very careful version control so you can show the journal what was the study protocol at the initiation of the study, is the place we look for the eligibility criteria, the randomization procedures, the treatments, and the primary, secondary endpoints. Okay? So obviously, we're going to look at the study protocol and you want that to match your manuscript. So the lesson here is just basically take that structure and put it in your manuscript, shortening it for the appropriate format. But there also is going to be a separate statistical analysis plan in most cases. And we look there to make sure that the primary and secondary endpoints haven't changed. Sometimes the SAP gives details about the definition of the primary and secondary endpoints that are actually missing from the protocol that are very important. What did you do in terms of defining a treatment success if the patient was missing one of the component variables, for example? We look to see what the SAP says about the exploratory endpoints, clearly the statistical analysis model and interim analysis plan. And sometimes authors have separately published, and I think this is good practice, to separately publish a design manuscript that lays out the design of their trial. For example, the journal Trials publishes quite a few of these. And that sort of publicly posts what they're thinking about the trial was, usually prior to its initiation. There's also a DSMB charter if you have a separate data safety monitoring board, which sometimes has additional information about the interim analysis plans. And I should have mentioned trial registration. Notice how I have trial registration and the design publication in the same sort of column. They're both pre first patient in, ideally, records of what you were thinking when you started the trial. And therefore they should agree with and complement each other. I will tell you that very often we find that the trial registration, which is required for submission to major journals, is done very early, and then it is not updated as changes are made to the trial. So it's really important if you have a major protocol change and you change, the definition of an endpoint or the ordering of endpoints, you go back and you fix your trial registration so that it has an audit trail as well that matches the evolution of the rest of your documents. One of the things that we look at a lot in the manuscript is the consort diagram, which tells us the degree with which the patients were followed as intended by the protocol. Again, the reporting of patients being lost to follow-up or having missing data ideally would be clarified in the statistical analysis plan, meaning how it's going to be done. We want to see that in the manuscript. So my point of this slide is that you want to make sure that not just your manuscript is in good shape when you submit it. You want to make sure that you've taken good care of document hygiene and version control along the way, so that you can submit all of these documents. Apologize for that. It is much better to submit all of these documents with your original submission than to wait to see whether the journal asks for them. Because what this does is it just demonstrates that you had your house in order, and that makes a good impression. And that impression really can affect the decision the editors make regarding whether to request a revision. Okay. So what are some self-inflicted injuries, meaning things that people do that hurt their chances of publication that they just didn't have to do? So, treatments sometimes don't work, patients sometimes don't show up. Things like that are unavoidable challenges. And actually, journals, almost everybody who's involved in journals has done research themselves. They really understand those things. But you want to avoid the self-inflicted injuries. So obviously inconsistency across study documents in the manuscript, and just take the time to make sure you've worded your definitions of all of the endpoints and their ordering exactly the same. Don't have it be 90-day survival in one and 60-day survival in another, and you would be amazed at how often that happens. So version control issues. Um, Once you have access to your data, any change you make to the analysis plan is suspect, meaning the thought is maybe you peeked at your data and decided, hey, this is the analysis I wish I had done because it makes my treatment look better than the analysis I pre-specified. So you want to make it unambiguous which changes to your analysis plan were before you had access to the data, maybe before first trial in or before locking of the database, and which were afterwards. This does not mean you can't make changes to your analysis plan after you have access to your data. Sometimes, for example, a manuscript will say, initially we had no plan for handling missing data. That'd be kind of a misstep. But because there was 10% missing data in this endpoint, we've decided to use multiple imputation with, this technique, this many chains, in order to, account for the missing data. You've told us why you made a post hoc change. The change that was made is a typical approach as to one that looks cherry-picked to get the answer you want. That's largely going to be okay. Also, failure to include all elements required by reporting guidelines. You need to pay attention to consort reporting guidelines and make sure that you either give everything that's requested or explain why it's missing. Sometimes there are changes between initial and final design details, analysis plans that are we just find digging through the documents, and the authors never mention why they did something. And I'm talking about things like dropping doses, changing inclusion criteria. I mean, substantial changes. Things like that should not go unexplained. Another issue is representativeness of the study population. We were just discussing a manuscript yesterday where the distribution of sex in the enrolled population is different than the known distribution of sex for the target illness. And that's odd. Why was there apparent selection based on sex? The clinical importance, validity, and patient-centered of the primary outcome. Sometimes primary outcomes are chosen based on either historical practice or because the investigator or the sponsor thinks it's the thing that's most likely to respond. But you step back and you say, this isn't really what patients with the target condition really care about. Another issue that comes up is co-primary endpoints. So I will tell you that I find the term co-primary to almost always be a red flag because often academic investigators think co-primary means two endpoints, and if either is positive, it's a positive trial. The FDA thinks co-primary means two endpoints and both have to be positive for it's a positive trial. So co-primary in an FDA setting means two chances to fail. In an academic setting, people think of it as two chances to win. Either might be reasonable designs, but you don't want to leave it ambiguous regarding what you mean. Other issues is are overly optimistic assumptions. For example, an underpowered negative trial with a trend towards benefit is not going to be one that's easily interpretable or can be communicated to a clinical audience. Um, and then poorly defined analysis models. How exactly were you going to estimate the treatment difference? How are you going to deal with missing data, intercurrent events such as being lost to follow-up, deaths, competing risks, for example. Another issue, and this is just a laundry list of things that come up, and it's just something to look at when you're preparing a manuscript. Saying you're doing a modified intention to treat analysis without a clear and precise explanation of what is different from ITT. As you know, intention to treat means patients are retained in the treatment group to which they were originally assigned, regardless of what happens to them. Give me just a second. If you're going to modify that, how are you modifying it? A common modification is that you only include patients who receive at least one dose of the study drug, placebo or active. That may be reasonable. Don't call that modified intent to treat. Just say we included in the analysis population these people. Justification of non-inferiority margins is a very challenging area. A common strategy of simply saying 50% of what the treatment effect of the active arm is not a very good justification. This really needs a clinical justification based on the presumed advantages of the experimental agent. We do non-inferiority designs because we think the experimental arm has other advantages. You need to make an argument about how those advantages compare to potential loss in efficacy in the primary endpoint. Handling of missing data, management of type 1 error control when you have multiple endpoints you're testing. For example, if you're going to use a gatekeeping strategy, you actually have to lay out the exact ordering that you will in which you will test the primary and secondary endpoints. And then clarity of any interim analyses. What was the plan? What was actually done? In study conduct, so those were all design issues. In study conduct, it's really adherence to the study protocol. Did patients receive the intended treatment? Did they follow up? If you have interim analyses, and especially if a decision was taken or made at an interim analysis, was it an ad hoc decision or was the plan both well laid out and followed? During analysis, you want to first perform your pre-specified analyses, and then if there's any other analyses, you want it to be clearly laid out what the post hoc analyses were and why they were performed. The use of per protocol or as treated analyses. So this is, I don't want to use alternatives. These are, I want to say the opposite of intention to treat, where you include patients in treatment groups only if they followed the protocol or according to the treatment they actually received. For example, if there were crossovers, those are rarely useful. The exception is that an as treated analysis or per protocol may be helpful in supporting the ITT analysis in a non-inferiority trial. And then in settings in which you need to be adjusting for confounding using modern statistical methods, such as propensity scores or inverse probability weighting or target trial emulation. If you're in one of those settings because you're using an observational data set, failure to do that is a real problem. Sort of naively analyzing observational data as if they had come from a randomized trial is problematic. This is a bit of a fine point. I just want to say where we see that is where the data were from a randomized trial, but people are using those randomized data to evaluate the effect of a different treatment that was not the randomized treatment. That is an observational data set, even though it arose in an RCT. and you want to be using appropriate techniques matched to the observational setting. And then, as I mentioned before, post-randomization exclusions. In reporting, and I think this risks getting redundant, it's clear and transparent reporting of exactly what happened and according to the consort guidelines so that no patients are lost. And then the interpretation, I just have to say, it is much better to be your own critic and be measured in your interpretation of your data than to have the reviewer have to be the one to point out that you're overinterpreting your data. Don't try to spin a trial with a negative primary result into a positive result. If there are positive or suggestive things within secondary endpoints or analyses, it is fine to point those out and to place them into context in the discussion, but do not try to turn them into bottom line results in your abstract or your final conclusion statement of your trial. Okay, that makes it look like you're trying to spin. I think we've handled these. Then there are other things that people sometimes ask me, are these okay? Do journals um, look down upon these kinds of approaches? And I just want to say that all of the major journals um, have become substantially more methodologically sophisticated over the last decade and are now used to seeing things like adaptive trial designs, factorial trials, cluster randomized trials, pragmatic trials, use of Bayesian methods. I don't think any of these things are a red flag for the higher quality journals at this point. But for each of these approaches, all of the stuff I've said earlier about pre-specification, being clear about what you plan to do, what you did, why you did it, and when there were changes, why there were changes, apply regardless of what the design is. For example, a pragmatic trial design is not an excuse for a lack of clarity regarding your inclusion criteria or the treatments or the endpoints or the analyses. All right. So let me stop there. I'd be happy to take any questions. So I will turn it over, I think, to Megan.
Thank you, Roger. That was excellent. And we are looking forward to some good discussion. So please, if anyone has a question, feel free to raise your hand. And you can ask your question out loud, or if you type them in the chat, I can help facilitate.
I wish I had met Roger 20 years ago in writing papers.
I'm gonna have kind of a
Thank you
question
for
for Meg.
clarifying the context, Mark. Go ahead.
I'm Daryl Miller. I'm from Indiana University, and I am honestly, I've never done a clinical trial ever, and I'm actually trying to work on getting funding right now. Is kind of where I'm at in my career, so I'm very ignorant, and I've never heard of publishing a trial plan before. Like, is that a good idea to do even before you get funding? To like say, hey, this is what I want to do, and I'm going to publish explicitly, so that way the people reading through your grant, even at that stage. Like,
Yeah,
because that would
absolutely.
probably keep things really consistent
Yeah, so
throughout.
absolutely.
And then, can you tell me just a little bit more about that? You mentioned the journal name, but I think I missed it. I'm so sorry. I'm just curious.
Okay, sure. So first, absolutely, it's a good idea to publish a design manuscript if you can, and even before you submit a grant, because you can put a lot more detail into that manuscript than you can into the 12 pages, for example, you might be allowed in your grant application. So the journal I mentioned, and I didn't mean to, endorse a particular journal, but it's just called Trials. Okay, it just happens to publish a lot of these types of design manuscripts. The purpose of doing this is it first of all, it draws a line in the sand. It says, as of this date, this is what I envisioned for my trial, and therefore you can think of it as a highly detailed clinicaltrials.gov registration. But you can include as supplementary materials a statistical analysis plan. You can include your clinical protocol with all the details. And even though some of these things may change, and we all expect them to change, it gives a complete starting point. And as a reviewer of grants, if someone says the details of the design have been published, I am not obligated as a reviewer to look at that, right? I can review to, I'm only obligated to read the materials you submit that are allowed in the grant. But I'll tell you, I will. And the statisticians and the methodologists will, and they'll look at this, and that will either increase their enthusiasm for your project or decrease it, depending on the quality. But the upside is much bigger than the downside. And very often, the people who just read the materials you submit will say, it was really unclear how they were going to handle this. And if someone could say, yeah, but if you look at the design manuscript, they really did lay that out quite clearly, and there was a protocol, and this is how they were going to handle it, that can really help you during peer review of your grant. So I'm all for it. It requires planning and time and effort.
Great to know. So just got to do a good job. That's the truth.
Robin asked, when one should submit JAMA versus JAMA Neurology, or would the authors be redirected? So JAMA is a general medical journal, so it tries to publish things that wide groups of clinicians will find interesting, although there may be such important advances that are in a narrow field that we think they're appropriately published there. JAMA Neurology is, not surprisingly, focused on neurology. So if you have something that you think is really of interest to neurologists, it probably makes sense. I mean, with somewhat of a more narrow focus, it probably makes sense to go directly to JAMA Neurology. But you can submit to JAMA and at the time say that you would allow an internal referral from JAMA to JAMA Neurology. And what happens is that that initial reviewing editor, instead of just rejecting outright if they didn't think it was JAMA, will send it on to JAMA Neurology. And there's no penalty or negative connotation to this. And commonly what will happen is it'll be sent out for review at JAMA. We will do our methodologic review, we'll do our content reviews, and we may find that it's just not a good fit for our readership, but we think it's important. And we have lots of neurologists on our editorial, in our editorial group. And what we will do is we'll send it on internally with the author's permission, if they've pre-specified, to JAMA Neurology with our reviews. So there's actually not even loss in time. And then JAMA Neurology will make its own editorial decision based on their areas of emphasis. The other thing that one can do is one can simply ask. One can send a near final draft manuscript to the manuscript submission email at JAMA and say, do you think this will be better in JAMA or JAMA Neurology? And believe it or not, a very senior editor will look at it and give you advice. People don't ask for advice enough. Everybody's trying to do the right thing, so they will help you try to do the right thing. But as a general rule, I would, if you think it has a chance at JAMA, I'd send it there with specifically checking the box that says, if not selected for JAMA, please refer to JAMA Neurology. Other questions?
We have time for one or two more.
Maybe rather than having co-primary as well, because it sounds like that's a pitfall, is it better to just have one primary? And I don't know, I'm just gonna throw it out there, 5 secondaries. Is that more clear? I don't know. I'm just reading through things that maybe that would make sense.
So I think that
what do you do if you think things are fairly important, but you can't quite make them co-primary, I guess is what I'm asking about.
Yeah, so this is really a question about the clinical context or the scientific context for the endpoints. The two uses of the term co-primary reflect completely different scientific goals. So the co-primary where what you want is if either of these positive, I want to call it a positive trial, where you have to split your alpha generally between the two. That's the setting in which if their treatment worked on either of these outcomes, we would care and we would use it. So if it, improves survival or improve quality of life, either of those would be important and we don't require both. Sometimes the FDA is in the opposite setting, or sponsors, where you actually need both of them for it to be accepted. So one might be a safety and one might be an efficacy, and unless it's both safe and effective, we're not interested in it as a new treatment. So then it would be the different use of co-primary. The third case is one where you really have one thing you're most interested in, but if it worked on this first endpoint, you'd also really you'd want to know about these other things. So for example, in a chemotherapy setting, you may want to look at disease-free survival. But if you had benefit in disease-free survival, you'd also be interested in the burden of, say, of certain, very difficult to handle, complications. And in that case, that's more of a primary than secondary thing. So I think the question is, what endpoints do you need to show movement on in order to propose the treatment as being an advance? And then the secondary endpoints are the things you'd also like to know about if you were making a decision to adopt the new treatment. So not particularly clear. I'd be happy, in individual conversations with the specifics to help give guidance on how to order endpoints and how to label them. I will say, as my last comment, there has been a trend towards having an unbelievable number of secondary endpoints in protocols. Okay. And that is sort of disingenuous. Calling it the 8th secondary endpoint does not make it more important than calling it an exploratory endpoint. Just means you couldn't make a decision. So I think there should be usually one primary and a very small number of secondaries. Think 5 or fewer. So
Good to know. Thank you.
Yep. Thank you. People want to email me questions, happy to answer, and I look forward to seeing everybody face to face.
Thank you so much. Thank you, Roger, for presenting, and to everyone for attending.
Okay. Thank you. Have a good day.
Soon.
Thank you, Roger.